So everyone, welcome to the third Biocentrica Presents. Tonight we're joined with Roy, who is a clinical pharmacologist. She's going to be taking us through what goes on in clinical pharmacology. As a soft reminder, if you have questions, please hold them for the end when we have our speaking component, or if you would prefer, you can type in your questions into the chat as we go along, and I will read them out in a Q&A style format at the end to Roy. So without further ado, Roy, please take it away.
All right. Hi, everybody. My name is Roy, and like Penny said, clinical pharmacology I love absolutely, and it's a very, very fun field, in my opinion. So I'm hoping to sort of let other people know what sort of things clinical pharmacologists do, what sort of people also work within clinical pharmacology. So moving on, just as the housekeeping rules, audience guidelines, etc., I know that right now you all are in attendance, so there shouldn't be any noise as on your end. If you have any questions, just type it into the chat as well. Okay, disclosure-wise, I don't have anything. This one is going to just be an overview of clinical pharmacology and a little bit about me and my background. So I started out actually in general biology. That is what I got my degree in. And I did an engineering degree in the beginning, but after attending a symposium, I thought, oh wow, pharmacy is actually a very interesting field. And I was interested in the fact that you could do a lot of things. So you could enter the Air Force, go to retail, or you could do industry, hospital, etc. So to kind of catapult myself, I started as a research assistant in cardiology. And I was also a pharmacy assistant, and then I was also working part-time as a content operations manager just for a tech company during my time in school. Then I moved on to getting my pharmacy doctorate, and this I was a lab assistant as well in a pharmaceutics lab. I ended up getting a paper published as well, and I had done investigational drug services. So those of you who are P4s, in case you are ever so interested, or P3s actually, if you're interested in industry and you're not able to get an actual industry APPE, investigational drug services is quite a nice area to go into because you will be able to see all the protocols, the investigational brochures. You'll be able to read all of them, and then you will see on the back end of how do pharmacists, check over the medications, check to see right patient, right drug on that end. And so there's quite a lot that goes into it. And I would strongly, strongly suggest that you all check it out. I was also a compounding pharmacy intern. This was just at a regular community independent pharmacy, so not hospital compounding. And then I also did an internship during my time, and it was in drug product development. After I graduated, I ended up becoming a clinical pharmacology fellow, and I had therapeutic areas in opioids and antipsychotics. Ended up getting 2 papers published in my fellowship, which was really fun. And I absolutely loved my time there. But of course, I need a job. So in the end, at my internship site, my previous manager invited me back. He just asked me, do you want the job? And I ended up getting a job as a drug product development scientist. This is a huge pivot from what I was doing originally because drug product development is within the CMC space. So those of you who don't know what CMC stands for, it's chemistry, manufacturing, and controls. This is the area where if you're interested in manufacturing, active product ingredient, formulation development, analytical development, those are more geared towards CMC, okay? And over here, the fun part about it is that I worked in different therapeutic areas of immunology and oncology. After that, I decided to go become a clinical pharmacologist again because still my bread and butter and something that I understood quite well. But this time around, I only focus solely on oncology. Immunology is a lot of fun, of course, but I think oncology is equally fun, if not more fun, just slightly. But with everything that's been happening and everything, I did recently just get a new job as a clinical scientist. So yet again, another pivot. And I think that's the fun thing about clinical pharmacology is the fact that you will still see what a clinical scientist does. You can do what a clinical scientist does. You just need to understand the way that industry works. And I think that is one of the highlights of industry. Once you start to understand, once you get your degree and you understand the science, now it just becomes understanding the role. And so that's why I was able to pivot into clinical science and to top it off in a completely new therapeutic area of endocrinology. So a little bit about the landscape of clinical pharmacology. What is it exactly? And so this is an integration of PK/PD, so pharmacokinetics, and then pharmacodynamics, ADME, which is absorption, distribution, metabolism, and excretion, and dose selection across all development. So clinical pharmacologists do not just sit in early development. Okay, we have all of the PK, we don't need them anymore. That's actually not the case. They go through all the way from phase 1 all the way to BLA NDA filing because there are very, very many studies that clinical pharmacologists need to also do. You will also evaluate something called exposure-response relationships. So this is, in a simple way, it is essentially as the drug increases, aka as your exposure to the drug increases, do you have an increase in response? And these responses can be either safety or efficacy. So you usually have many graphs, etc., that are portraying this particular exposure-response relationship. And the optimal dosing strategy for diverse populations. When we're talking about diverse populations in industry, we're thinking pediatrics, geriatric, hepatic, renal impairment patients. And there are other ones, but those are usually like the hard-hitting ones that you will see a lot more of. Regulatory submissions with modeling, simulation, and justification part. Regulatory submissions, like I said, you are part of it. You have your own specific section within Module 2 that you do write as well. And that, I think, really lends itself to how much clinical pharmacologists are very, very well regarded in that space. Okay, so now we're going into a little bit of where you can go for the practice areas. This is not all-inclusive. I want to point that out right now, but there are 4 that I really think are going to be something that you, if you're interested ever in clinical pharmacology, will hear these terminology a lot. So there's ClinPharm, pharmacometrics, physiologically based pharmacokinetics, or better known as PBPK, drug metabolism and pharmacokinetics. This is DMPK. Okay, so clinical pharmacology, you can kind of think of them as the equivalent to the clinical scientist, but in a very, very niche area, which is only clinical pharmacology studies. So they are usually the ones who will deal with the first-in-human studies, aka phase 1 studies, SAD and MAD, which is like a single or multiple ascending dose study, food effect, DDI studies, hepatic, renal impairment studies. These are all under the umbrella of a clinical pharmacologist, and they should have a lot of ownership alongside of their clinical scientist for these protocols and for the synopses and investigational brochures. You really want to be able to determine dosing regimens using the exposure response data. You can assess the PK differences and then the reviewing of the clinical pharmacology sections like what I mentioned, specifically module 271 and 272. You'll have, because you work a lot with clinical scientists, that means that also comes with the territory of working with regulatory folks and just a very cross-functional team in order to let them know this is why I chose the dose that I chose. But not only that, but now you need to talk to your CMC folks, right? To let them know, this is the dose we're choosing. Can you make me a formulation specific for this dose that we're choosing? And so that's what I kind of mean by this cross-functional discussion. It's extremely important that everyone is really kept in the loop based off of your decisions. As well as collaboration, like I mentioned, right? Clinical, regulatory, you have your bioanalytical teams, and you want to be able to evaluate and interpret these data, okay? So they are kind of the overarching umbrella, because then when we go into pharmacometrics, they're kind of your support, okay? They're the ones who are really going to be in the nitty-gritty hard sciences. And when I say hard sciences, I'm talking about they're making the models for POP-PK, they're making the exposure response models and the time to event models, okay? You, as the clinical pharmacologist, you will evaluate and you will interpret said models. But most of the time, that's what the confusion kind of comes in. You can become a clinical pharmacologist without being a pharmacometrician. And they're just very, very heavy in the nitty-gritty of like learning how to make these models. So you can do justifications for dose selections, your special populations, etc. You obviously contribute to your filings. This is all also under the guidance of these clinical pharmacologists. And you can translate these models right into insights for regulatory submissions, and you can be responsible for a lot of those bridging strategies across global regions. Now, the next one, they also kind of work in tandem with pharmacometrics. But these people, these PBPK people, they're the ones who model specifically drug absorption, distribution, metabolism, and excretion. Again, they are a very big modeling group as well. And so they're using human PK data and preclinical data and in vitro data, all of this data in order to create your model. So a lot of the things that they actually look at are DDI potential. So let's say you have a drug-drug interaction study. Usually you will use a strong inhibitor.
Roy, are you there? Oh dear. Can you all hear me? Okay, I think Roy may have disconnected momentarily. Uh-oh. One second. Let me...
PK.
Oh, Roy?
Oh, hello?
Hey, sorry to interrupt your presentation. It seems we lost some sound for about maybe a minute there.
Oh, okay. So where did you leave off on, actually? Did we finish this?
I think we were on... Chat, help me out. PBPK. Right, the strong inducer.
Okay.
So second to last
perfect.
bullet.
Okay, so it's very, very important for like the PBPK folks, because if you have a strong inducer or inhibitor, let's say, and you have information from that for what the exposure response was going to be, now these models can actually model what would happen if you have a moderate inhibitor or inducer. And this will really, really help your regulatory submissions, so then you don't have to actually do a second study specifically targeting those moderate inhibitors and inducers. Of course, these folks are also able to model if you have a hepatic impairment, renal impairment, and also, like you may know, there are different stages to how bad your impairment can be. So again, they can model all of that information. Okay, the last one that I want to talk about is DMPK. DMPK, in my opinion, is sort of like the beginning stages of what is going to happen. They're the ones who work a lot more closely to non-clinical. And when I mention non-clinical, I'm talking about the people who do the animal studies, the animal models, essentially anything that is not in a human. They are going to create equations, models, etc., of what happens from going from a animal model to a human. They're the ones who will decide, most likely, what is going to be your first in human dose, for example. You'll be able to see what sort of enzymes are involved based off of, right, your in vitro studies or your in vivo in animals. And they'll be able to look at all of the ways that the drug is going to interact in metabolism terms with your body. So they're trying to mitigate any risks, right, related to your DDIs, any bioavailability risks, and clearance mechanisms. So they're very important in the very beginning stages, right, when you're going into a human for the very first time, because you want to make sure you're not going to overdose them. It is okay if you underdose, of course, because if you underdose, then that's essentially the point of your first in human dose. You want to be able to slowly make your way up to find what that efficacious dose is. Okay, now we're going to move on to responsibilities. Responsibility-wise, of course, you're going to have study design responsibility. There are a numerous amount of clinical pharmacology specific studies that you will be in charge of, like what I mentioned, the DDIs, renal hepatic impairment studies, those are the big ones, as well as any ADME studies and mass balance studies. First in human is also usually in the wheelhouse of your clin pharm team. The second one is the modeling simulation. So these folks are the ones that I mentioned previously, your pharmacometricians, PBPK, and DMPK folks. Labeling support. So this is actually going to be the entire team. You need to have a very strong labeling support team and say, this is what the clearance is of the drug, the half-life, etc. And these all need to go into the label. You also need to have summaries of what exactly happened in all of your studies. Is it okay taken with food? Or is it not? Is there going to be, do you want to take it sort of without food or with food? And if you can take it, for example, grapefruit juice, any inhibitor or inducer. Regulatory interaction, of course, you will be attending a lot of FDA meetings if it's a clinical pharmacology relevant one. A lot of the times it will be end of phase 1 meeting, end of phase 2 meeting, and also your pre-BLA, pre-NDA meeting. Those you will very commonly see your clin pharm folks also attending. And the last thing is the bioanalytical review and your table listing and figures. That's what TLF stands for, quality check and quality control, I mean. So, and okay, of course, there is a lot of skills for clinical pharmacology. So I sort of bucket it into 3 different sections. You need to have the scientific and technical knowledge. Obviously, if I'm going to be saying all of these acronyms, for example, these are actually the bread and butter acronyms. And so you need to understand, how does the drug work on a body? How does the drug work in terms of, is it going to harm a patient? Is it going to help a patient? Why is it going to harm them? And why is it going to help them? These are all things that you actually need to understand very, very well, because you cannot model and you cannot simulate something if you don't understand what you're modeling and what you're simulating, right? The next one is tools and platforms. There are a lot of tools, a lot of modeling tools, a lot of things that you can use to do data analysis, create graphs, etc. You're going to be making a lot of graphs. You're going to be analyzing a lot of data. And so this is not an all-inclusive list, but I want to say these are some of the most commonly used ones, at least, for tools and platforms. And you also don't need to know all of these tools and platforms. Usually a company will have 1 or 2. So just as long as you know the most popular ones, you should be okay. And as for soft skills, I think actually soft skills for clin pharm is probably the hardest, in my opinion. And that actually goes for many, many functional areas. Soft skills is something that you can't really learn in school. It's learned on experience. And you have to know scientific writing. You have to be able, because, right, a lot of this is scientific and technical knowledge, you need to be able to essentially tell other people, what does this information mean and why is it important to you as an audience? So being able to just communicate that to non-expert audiences, of course you want to be able to have that attention to detail because you're doing data analyses, right? It's very, very data driven of a functional area. So being very data, having that attention to detail will definitely help you in the long run. Time management, comfort with ambiguity, because like I mentioned, the exposure-response relationship, a lot of the times, sometimes if it's like a flat one, then you need to be able to sort of make the decision or the call. I actually do think the higher dose is better because of X, Y, and Z. Or you have to accept the fact that the lower dose is going to be better. But sometimes, honestly, there's just no right answer. And you need to be okay with that. And that is a lot of the times in clinical pharmacology. Of course, with these comes a lot of challenges. You will get a lot of incomplete and variable clinical data, especially the smaller your population is, the smaller amount of clinical data that you'll have, and that increases variability. So this kind of goes back to statistics, where You want to have small standard deviations, small standard errors, because you have a big enough population. The second one is tight timelines for deliverables. Clinical pharmacology cannot work until everything has been locked. And when I say locked, I mean things like database lock, where, okay, all of the data is in, all of it has been verified by our data management team, and no one can touch the data anymore. That's when clinical pharmacology comes in, makes these graphs, does the data analyses. But the turnaround time is very, very fast, especially if you're at a pharma company, because you have the data. The question becomes now, why can't you generate the outputs faster, right? And so you will a lot of the times have some late nights, yes, but I think it's quite rewarding at the very end when you see all of your data, just being able to understand your data and understanding why you made some conclusions. And like I mentioned on the previous slide, you will have these data interpretation challenges, like I was mentioning with that exposure response. Is the higher dose better? Is the lower dose better? Sometimes it's not very clear. And a lot of the times there's going to be communication gap. Because this is such a technical role, sometimes the message gets a little bit lost, or clinicians might not understand why is it that we care about these exposures? Why is it that, we actually have a pretty good safety profile, can't we just go straight to the higher dose? But honestly, that's not the point of going to the highest dose. You're trying to find a dose that is not only safe, but effective at hopefully the most minimal dose that you can have. And that's where a lot of these communication gaps and challenges are going to come in. Okay, now moving on to everyone's favorite part, which is education, career progression, and salary. Education-wise, we'll have our 5 on the very side. I will say that I don't think I've actually ever worked with a bachelor's or a master's degree. None of my companies have a bachelor's or a master's degree in their clin pharm department. So that is why I'm saying you have to be lucky, like very, very, very lucky. And know exactly what it is that you're getting into. On the other hand, of course, PhD, PharmDs, MDs, you need a doctorate essentially to get into this field. And that is a very common thing. Most likely than not, you'll actually be seeing a lot more PhD. So PhDs in pharmacokinetics, drug metabolism, pharmaceutics. These are all very common PhD programs that you can enter. And that's where you're going to see majority of clinical pharmacologists. I think nowadays the trend is moving a little bit more towards having some PharmDs running around. We have some fellowships as well, but it's still early in the process, so you still won't be seeing too many of them. Okay, career and salary progression. So for clinical pharmacology in general, you will be an associate level unless you go into a fellowship. The fun thing actually for these postdoc fellowships is the fact that because there's so many PhDs, you will likely be able to go directly into industry. You do have a higher salary, okay, as a fellow, most likely than not, unless you go into a PharmD specific fellowship, okay? And that is the difference. So if you want to do a PharmD fellowship, you will get, right, that 50, 60K as, part of Rutgers MCPHS. But for postdoc fellowships, like at Merck, J&J, Genentech, etc., you'll likely be able to clear around 100K easily, like straight out of school. And you're still in a fellowship. Then you kind of move on into your mid-level career. You'll start getting that scientist title, senior associate. You might be a manager, depending. You probably won't be able to manage anybody at this stage, but you'll have a manager title. It's very interesting. And your salary will only go up from here. Right, so 115 to 150K. After that, you can now start thinking about your mid-level. This is where you're going to see a little bit more associate director role, senior manager. Some places do still have mid-level as a senior scientist. So just be cautious. Just look at the years of experience. That's what mainly is going to help. And then your salary is going to, again, go up again. After that is going to be the leadership. So you're looking at directors, group leads, etc. And salary, again, you'll increase. The nice thing about clinical pharmacology is that there's usually no limit to what you can do. And that goes for a PharmD and a PhD as well. You're not usually competing that hard anymore once you get towards these leadership roles with your PhD fellows, colleagues, when you're a PharmD. Once you get to, that advanced mid-level role, they expect you to essentially work at the same level as a PhD, which is great. And then after that, senior leadership. This is going to be the area where work-life balance will be a lot harder, to be honest. And that is just to see how motivated you are to rise up all the way over there. And this, honestly, this salary has no cap at that point. So hiring-wise, what do we care about when we're looking for a clinical pharmacologist? There's going to be a lot of information on these slides, by the way. So bear with me over here. Prior experience-wise, it's kind of questions, I think, that you might want to just ask yourself as you're going through a hiring process, right? Have you done any protocol development, PK, regulatory submissions? How familiar are you with all of these studies? Have you led any studies? That's pretty important. And have you worked anywhere? The fellowship route is a perfectly fine place. If you do a postdoc fellowship, a lot of the times in clinical pharmacology, they will count your fellowship experience as years of experience worked, by the way. That's also a positive. And can you interpret models? Can you understand what these model outputs are even telling you? And have you ever done cross-functional study teams? The next one is your scientific acumen. You need to understand what is going on. Okay, so can you talk about PK/PD concepts? Are there a lot of dose exposure responses, bioanalytical data, regulatory guidances? There's quite a lot for clinical pharmacology, and you will find a lot of it on the FDA site. ICH guidelines also has quite a bit of things that you can read. And then can you write out, review these sections, and do you understand that trial design? The last thing, again, is the soft skills. Soft skills-wise, communication. That is very big. Can you communicate your science into something that's easily digestible? I'm just going to hone in on that over and over and over again. And can you do these timelines? Can you shift your priorities? What if your higher dose doesn't actually work out? Can you shift your priority to the lower dose? Can you shift to a different dose? How flexible can you be? And then feedback is always very important. This honestly goes across all functional areas, not just clinical pharmacology. And collaboration, dependable, curious, and definitely easy to work with under pressure. So a little bit more about our wrap-up slide. So breaking into the role a little, I have reached out to clinical pharmacologists as well on LinkedIn or at conferences. I feel like referrals do go a long way. During my job hunt, I did get quite a few referrals, and it was mainly the referrals that did give me at least a first-round interview. You want to be able to help other folks understand why you're valuable. You want to be able to tell them, hey, I understand your data. I can collaborate. I can distill my data into very, very simple slides. And demonstrating that communication, okay? This is very key because I'm not going to say across the board, maybe a good chunk of clinical pharmacologists, because they're so entrenched in the science, sometimes communication skills might need some work, but being able to communicate, I think at this part, it will definitely set you apart. The second thing is there anything you can do right now, right? Can you join memberships, training? I actually really like clinical pharmacology conferences and everything, and their membership, because if you're a student or trainee, it's really cheap, if not free, to get a membership. Just FYI. And I love it because you can network. These conferences are going to be way smaller than the ones that you're usually hearing about on the news or where there's a lot of data readout, etc. This is just essentially, to me, networking opportunities, and you'll see the same people at these conferences over and over and over. There are some pharmacometric certifications you can get online, and the Phoenix training courses are actually really, really well done. So, but it does cost money. So see, if your school offers them, etc., and see what you can learn from Phoenix. And I'm sure that there are YouTube videos out there. It's just Phoenix itself costs money. So it might be a little bit difficult to follow along sometimes if you're more of a hands-on sort of person. The last thing is building your portfolio, right? You want to be able to contribute to study designs. So like I mentioned, when I was in pharmacy school and I worked in a lab, I was the one who was getting informed consents from a lot of people. I was also the one who was doing a lot of study design. How are we going to take these samples? How are we analyzing these samples? These are things that I really wanted to, put out there and tell people, this is what I am capable of doing. This is what I understand how to do. And if you don't have that opportunity, like I did, to do an actual study with a professor, for example, you can have documents, like just mock documents. There's like dummy data that you can always create, and it can help if you're trying to explain something, or even help yourself understand what exactly can this dummy data do? What am I even creating? Why do I understand this data? And of course, the last but not least is abstracts or posters at the conferences that I mentioned with those memberships above. And with that, now moving on to Q&A.
Roy, thank you so much. That was a fantastic presentation. I learned quite a lot from that, actually. So as you mentioned, we're going to move on to the Q&A. So for everyone in attendance, if you have a question for Roy, please post in the chat. I'll be reading starting from the top here. Are you ready, Roy?
Yes, I am scrolling right now.
Okay, perfect. Well, we'll start with the first one. Someone asks, how do you foresee the clinical pharmacology job market changing in the next 5 to 10 years?
To be honest, I have been seeing more and more people utilizing AI to do their models. Of course, it's not perfect all the time, but when it comes to like the nitty-gritty information of, just plotting, etc., like how do I even use this? Where can I get information on this? AI has actually been quite valuable in that sense. So I don't think that they'll ever be replaced. They really can't, because a model is only as good as whoever creates it, right? And there are so many variables. Every single human is different. So it's a little bit difficult, right? That's why we don't have a one-size-fits-all model yet. It's very hard to replace them. So that's kind of clinical pharmacology in general will, I think, be using a lot more AI. But I don't think the job market is really going to slow down for clinical pharmacologists. They're usually the ones that have a lot of job security. You won't really see them get laid off, usually, especially at a bigger company.
Perfect. Thank you. Next question is, which out of all of the tools and platforms that you showed off do you believe to be the most important to know, and why?
I love this. Okay, so tools and platforms, 100%. I really like Phoenix. It's a very easy tool to use. To be honest, it's like a plug and chug. I've used R, definitely. I think R is faster, but there's a huge learning curve to R. Okay, if you don't want a huge learning curve, you could just stick with Phoenix. Non-mem, that's a little bit more for modeling. So I'm personally as mem. I know non-mem is essentially a requirement for many job postings. So if you just do a quick LinkedIn, Google search for ClinPharm jobs, you'll probably see this non-mem word. If you have the time, if you have the capability, I personally would go ahead and learn it. And then the next one that's important would be Jump, in my opinion. Jump is like a nice little graphing tool, but can also do data analysis. So those, I think, are going to be my top, what did I even have? 3? Phoenix, non-mem, and Jump. Those are the 3 that I really like.
Gotcha. Perfect. And I have a personal question myself. Do you have to know any coding or have a coding background in order to use these tools, or are they mostly like Phoenix where it's pretty simple?
If you want to be a clinical pharmacologist, no, you don't really need to know a lot of coding. If you want to use R, that's essentially the only one that will require that coding knowledge. But like I said, it is way faster to use R. So if you know how to use R and you're good at it, you essentially are going to be a faster worker and it'll make your life easier. So knowing coding essentially just makes your life faster, easier, more efficient.
Gotcha, but it's not like
Not a
a hard requirement.
exactly.
Excellent, excellent. Our next question is, how often do you interface with other functional areas, and what do those interactions or collaborations look like?
Every single day. Like, you will talk to them every single day. I think the people who I usually talk to the most are going to be regulatory and clinical science. Those are the two that I would have a lot of interactions with. After that, it's mainly more on a higher level, like a higher overview, not so much of the nitty gritty information. You will have, trial team meetings where you'll meet with a lot more people, like clinical operations, etc. But those aren't going to be every single day. Like if you have a question, you can usually message them. I also do like clinical pharmacology because I personally think I work by myself a lot of the times to get my data outputs, right? Very quick and fast. And that's why I kind of like ClinPharm. It has that individuality portion of it, but then you can also reach out to other people and communicate, collaborate with them very easily.
Excellent. Timo asks, how has your background in clinical pharmacology enhanced your experience in clinical science and vice versa?
Honestly, I feel like they're pretty similar to each other. Like, just very, very basically very similar. You're still looking at trial design, you're still really looking at the protocol. You have a lot of similarities in terms of you need to wait for the data, essentially, and you're going to want to understand how a dose is very relevant to the patient. Oops. And I think that's kind of the biggest difference is clinical scientists will focus more on the clinical, what was it really safe? How is this efficacious? Is the drug even efficacious? That's a yes or a no question. ClinPharm comes in and tells you this is the dose we suggest because of X, Y, and Z. So they kind of like supplement clinical scientists in the fact that they're really trying to help them get to the right decision in a sense. And I think clinical scientists, they just have so much on their plate that sometimes it's really difficult, right, to be able to get every single piece of information. So that's why it's important for the clinical pharmacologist to distill information down to make it easy to understand. Some people just don't have the bandwidth or the time to look at this data nitty gritty.
All right, perfect. Our next question is, what's your favorite thing about presenting data?
I love it when it is exactly what you expected. Like, you say higher dose, better efficacy. Higher dose, worse safety, expected, within line. And I like it whenever it's just positive or negative correlations. And when you present the data, I like presenting data because you're presenting the data because it makes sense. You're not usually presenting data that doesn't make sense. There's always a reason, okay, not always. A lot of the times there is a reason why you're presenting and why you are seeing the outcomes. And I love being able to explain the why and then having people understand and be very excited and have that same excitement of, oh, yeah, I get this sort of feeling.
Excellent. I myself, I personally like to display the data, of course, as you probably know, within PowerPoint. So we're always thankful for every beautiful graph you produce. We love using it in our own assets, and it's just excellent. So let's see. The next question is, what type of challenges do you run into when trying to interpret data?
Sometimes the data doesn't make a lot of sense because there's always going to be outliers. So you'll usually do like a quick and dirty analysis, and then you'll start picking out the outliers because let's just say they had like a calcium excursion because of X, Y, and Z. You don't know what happened. And so there's like some of those. But in the very, very beginning, that's what the problem is. You need to sparse through all the data. You need to understand the data. And I think that's the challenge. Understanding why something didn't work or why something is running oddly, understanding why this particular piece of data is important. I think that's the biggest challenge because you usually you get like this huge data set. And when I say huge, I mean there's just rows and rows and rows for one patient. You need to be able to understand what is important in this data set and what do I need to graph. What exactly am I doing? Am I trying to get the mean? Am I trying to get the median? Standard deviation, standard error? There's just a lot of different things that you can analyze. So maybe a little bit of decision paralysis. And then once you get past that hump, I think it's usually more smooth sailing at that point.
Gotcha. And just curious, what's been your biggest challenge or most significant challenge in that regard? Like, what do you find to be
reviewing somebody else's data? Like, because you do not know sometimes what they did. They do have like a statistical analysis plan. They're supposed to have things nicely laid out for you. But the challenge is, how do I get the same answers as them, right? Or something close enough. Because there's a lot of things that can happen in the middle of data, and sometimes you don't do everything by the book. So I think that's usually my biggest challenge is reviewing somebody else's data rather than doing it myself.
Gotcha. Okay, Timo asks, and I think this is in reference to the previous Biocentrica Presents, do you like being the quarterback more or being the offensive coordinator?
I'm not going to lie, I don't know what an offensive coordinator is. I just know what the quarterback is, but I am not very familiar with football that much. So I will pass on that question.
That makes two of us. I'd try to clarify, but I don't know anything about sports either. So it looks like he's typing in the chat. Perhaps, do you like being double lift or freak?
That's even worse. I know that's a league reference, but I can't help.
I have no idea. So we'll come back to that question, I guess, if there is further clarification. But let's see. Eloise asks, are there particular certifications that might improve one's knowledge of the functional area and improve chances of success when job searching? Let's modify this. Anything beyond what you've already mentioned?
I feel like clinical pharmacology is a little bit harder because when I came out of school, I just learned everything from scratch. Like, I didn't do much certification other than the ones that I mentioned. So I might not be the best person to ask this. I know that Rick is here, and he could probably answer what he's done before in the past, but that for the certifications. As for improving chances of success when job searching, I feel like these are the skills, right? The slide that I'm on right now. Mm-hmm. Being able to show people that you even understand the basics. Because you can understand, because I think when everything comes down to it, for every functional area, you need to understand what exactly is this functional area. Why is this functional important? Because they would not have this functional area in a company if it was obsolete. But if you cannot understand where they fit within the company and at what stage, I think you will have a little bit of a difficult time interviewing, because you want to help people understand what skills that you're bringing to the table and what you are doing in order to learn about said skills, right? So I think just having that curiosity and having that good communication skill is quite important in clinical.
Okay, perfect. Let's see. I think someone. Okay. So L Chart has some series of questions. His first question: in this workflow, where he describes DMPK to PBPK to PM to ClinPharm, his question asks, where is the therapeutic window defined and where is it refined?
So I feel like therapeutic window, this one is going to be very drug dependent, right? You usually have, right, your first in human study. This is going to depend on safety. Once you finish that, then you're going to go on to phase 2. Phase 2 is where you're going to be looking at kind of the to see if this drug actually even works. So you'll have like preliminary activity data that you can have. This is sort of where you'll sort of define where that therapeutic window is. You're going to see lower dose, higher dose, where exactly does it fall into, can you have a range of doses, etc. And for refining part, I would like to think that the ClinPharm studies, so let's say that your drug is metabolized by 3A4. Okay. Based off of the information that you found in your phase 2, phase 3 trial, can you deviate from this dose? Okay. So for example, because of a CYP3A4, you end up getting more exposure to a drug. When you get more exposure to a drug, now you're worried, is this drug going to be safe for the patient? So that's kind of where that is this okay or not situation. But on the flip side, if your exposure is less, that becomes an efficacy issue. Will my drug still be efficacious at this lower dose because I've taken this CYP3A4? Okay. And so that's kind of where that refining process goes into, and it's like a continuous refinement all the way from phase 2 till submission.
So what happens then if you get to right before you go to submit and then you find that there's an alteration in that window?
You will cry first. And it is extremely hard, I think, to get to that point. Nowadays, I'm not talking about before. Before, I think it was a little bit better because we didn't have as many therapies out. But now we have like precision medicine. We have antibodies. We have things that can be very specific targets. So I think that it would be a company error if you went all the way to the end and not knowing this is an issue, essentially.
Gotcha.
Yeah.
L-Chart has a follow-up question. It looks like there are many tools, it looks like there are many established assays, tools, and packages for fitting, displaying, and interpreting models. Good thing we stuck on this slide. Yet these tools were not always around. How might a new technology be adopted in clinical pharmacology? What makes a tool useful?
I can only say things on my behalf. I think what makes a tool useful, honestly, is ease of use as well as the functionality. So it can honestly be the most basic things. The tools that I like to use are ones where I can change the font color, I can change the font size, but not just that. How easy is it to get a mean? Like, do I have to do a whole bunch of things just to get a mean? Can I just drag and drop? Those to me are very important because that goes into my speed. How fast can I analyze the data? And that's why I use specific tools. Because number one, I know how to use them. And number two, it just makes my work way faster. So for new technology wise, being adopted in ClinPharm, that would be mainly personal preference in my opinion. Like, it depends on what you like to use. It's just people have gravitated towards the same things because their coworkers use them.
Excellent. Thank you. Another question is, why can't you generate the outputs faster? How standardized are these outputs? Are they full unique bespoke reports, or are there effective templates that every therapeutic or therapeutic modality conform to?
So this is really difficult, actually, in terms of all therapeutic areas. I can tell you now, every therapeutic area is going to be different. Every single study is different. And the reason why is because your primary, secondary, and exploratory analyses are usually going to be different from study to study. There will be a lot of similarities, yes, but you're probably not running the same study 2 times in a row, right? You're maybe adding an extra arm, let's say. You maybe have like 50 more patients than you did in your previous one, or maybe even 1,000 more patients. So it just becomes the way that the study is designed. You can always improve too on these things. So when you look at FDA guidances, templates, etc., they do have actually guidances and templates there where they'll tell you, oh, we actually like things in this format for this table, right? You can use those, but the thing is, it's always changing. They're always trying to see what is a more effective way to read data. And you're going to have different personalities, you're going to have different perspectives. And so your graphs, tables, they're just going to change all the time. That's why I can't really say that they're standardized. I feel like every single year there's a revamp, if not every 2 years. You can maybe skate by for 2 studies back to back. But even then, you would then need to do those studies like in parallel or maybe stagger it just a little bit. So that's why I feel like it's a little bit difficult to standardize outputs.
Gotcha. Okay. And then LChart did have a follow-up to that previous question as well. And you touched a little bit on it with the FDA having them, but where else can these be found, these sort of template documents or any other resources in that regard?
So if you are willing, you can actually email PIs in like academic institutions and ask them, oh, hey, I looked at your paper and I was wondering if I could get the data set that you used to run. If they're nice, they will accept because they've already published their paper. If it's not a patent-related thing, it's not usually too much of an issue. And that could help you, honestly. And I know a lot of professors, genuinely, they really, really like when you're interested in their data. That's kind of why I ended up getting so involved into lab work, etc., because I just ask questions over and over and over again. And they just always want to answer because you're interested in something that they've dedicated years of their lives doing.
All right, fantastic. And so I guess just to kind of close out this section of these questions, using those data sets that are available, presumably from these PIs, you can use those to sort of fill in figures in other sections using existing tools, right?
Wait, could you repeat that?
I'm going to rephrase that because I'm not really sure what I said myself. So the second part of LChart's question was, are there available data sets, non-dummy data sets, that can be used with existing tools to fill in the figures and sections, I presume, of those mock documents? So perhaps taking existing data sets from PIs or something, wherever they may originate, and using those data sets to fill out sections and figures of those template documents.
Yeah, you could also honestly just play around with it. Like, how do you like your graphed, essentially? And what exactly from that huge data set that you can get your hands on, what exactly is it that you want to know? What answers do you want to know? Because there's something called post hoc analyses or ad hoc analyses. These are really fun because they're nowhere really in your SAP or in your protocol. You're just thinking about this after the fact. Hey, I actually realized Asian patients are doing a lot better than my Caucasian patients. So you do a subset analyses. And so these are the questions that you want to constantly ask. What sort of populations are these going to work in? Do they work better? Do they not work? Is it even statistically significant? I think these are really important questions when you look at a data set. So when you get that non-dummy data set, hopefully from a PI, you can play around, right, with R, Phoenix, if you have a subscription to it. Usually your colleges or universities will provide a subscription, by the way, just FYI. And so R also, by the way, is very financially friendly because it's completely free. A lot of these other things, they all cost money. And so that is also why a lot of people might know R and start out with R, because they're able to do their analyses for free when they don't have financial resources, essentially.
I think that's a really great point to bring up with the universities. I know when I was in school, I had reached out to our tech department to get access to SPSS, which, for the record, has been the worst program that I've ever had to experience outside of Mathematica. But I think it's a great consideration, students, if you have the opportunity to do so. Most colleges and universities, as Roy stated, do have those programs for pretty cheap as well. I think for SPSS, I paid $10 for a license, which is pretty crazy. Let's see. FDA asks Roy, what aspects of your work in clinical pharmacology do you find most fulfilling or exciting?
I think the data, and then having the outputs to the data, because everybody looks forward to that part, right? Once you get the data, everyone's kind of like holding their breath, asking, hey, did this drug work? Are we good to go? And I love that feeling. I love the fact that, the things that you are having an output for are things that people are going to be using. People are going to be basing a lot of their decisions on. And that's what I think I enjoy the most about that role.
And the pretty graphs, right?
Of course. The graphs are the best. I just have slides with only graphs. It's great.
Oh, I love it. As a medical writer, that makes me really happy to see because, not a lot of bullets, right? Hopefully your company colors paint the graphs very beautifully.
I will not lie. They're not the pretty. Like, they just kind of pick random color sometimes, so.
Oh, okay. That's not what I expected.
Yeah.
Let's see. FDA, in follow-up to a previous question related to working with other functional areas, he asks, which functional area gives you the hardest time?
Probably clinical science.
Really?
Because they're the ones who always want the data. Because they're the ones who you will usually be the ones presenting said data, or they need to be able to take everyone's data into account and then disseminate it, right? And so when I say they give you the hardest time, they're usually the ones who give you tight timelines. They're always asking you for information as you are doing the data analysis.
Gotcha. Maybe Timo can take notes. He's in the audience here. Okay, L Chart asks, in response to something you said earlier where you stated you have to demonstrate you understand the functional area. L Chart asks, in your experience, do large language models demonstrate the appropriate level of understanding of these topics, or does it not understand at the required level?
I think they do a really good job of answering your questions for basic ClinPharm things. But when it comes down, I think that's the fun thing too, by the way, about drug discovery and about pharmaceuticals, is the fact that a lot of the times you're not going to be working on something that has been approved. And you're probably going to be working on a different mechanism. And all of this goes into account for how your drug works. So that's why I don't think large language models are able to completely answer all of your questions. A lot of these mechanisms, well, nowadays, we're going more into proprietary information. They're not giving out their information like candy, so it's more difficult for LLM to learn said information to give you an accurate response. So that's why I find it difficult for AI to take over your job and to understand the topic completely. And that is why I don't think that they will be, not in the near future at least, be capable of creating perfect models as of right now, based off of the inputs that you give it or based off of the information that it already has.
I think we're starting to notice the pattern here where AI is not a replacement tool, but rather something for augmentation. And it's funny now that we've had our third Biocentrica Presents where this has sort of come up, and that's been everyone's sentiment. So it's interesting to me. I'm curious as to how it will go with future speakers as well, but just something interesting I noticed. Timo hits us with another analogy. So, in regard to clinical science and clinical pharmacology, do you like making the milk tea and tweaking the recipes, parentheses, clinical science, or do you like being the one who thinks up milk tea recipes that might be tasty, parentheses, clin pharm?
Oh, I'm always about coming up with the recipes because I know they will be tasty.
Well, there you go. All right, Christine asks, with regards to postdoc fellowships versus PharmD fellowships, aside from the pay gap, are there other standard pros or cons that make one fellowship more desirable than the other? Why might someone choose the lower paying PharmD fellowship over a postdoc fellowship?
I will not lie, I would choose the postdoc fellowship in a heartbeat. Because you are treated essentially like a full-time employee. You are employed by the company. You have a higher salary. Of course, that's important personally to me. And you are in no way really considered a fellow, but you also get the training that a fellow gets. Somebody might choose the lower paying PharmD fellowship. Number one, I think sometimes it is because of confidence. You're not just competing with PharmDs in a postdoc fellowship. You're competing with hundreds and hundreds of postdoc PhDs. And they're the ones who probably have a lot more of that scientific and technical knowledge than you do. And so that's why a PharmD fellowship might be a little bit more of an easier gateway to get in. But if you already have those skills, I do not see in any way, shape, or form a postdoc fellowship is not for you. I think that would be the way to go. And it's totally fine too if you wanted the PharmD fellowship. But I have a lot of faith, I think, in a lot of people's PharmD programs. And you can learn on the job. For people who are actually interested in clinical pharmacology, there's a reason why they're interested. And there's also a very specific type of person who goes into clin pharm. So if you already know you want to do it, then I don't see anything holding you back.
Excellent. Let me ask you this then. Let's say we've got a pharmacist or a PhD, vice versa. Have you, in your experience, seen like a PharmD applied to a PhD program or a PhD applied to a PharmD program?
I've seen both, definitely, and both will get accepted. That's not a problem. I will say that many PhDs don't know the PharmD programs either. And I totally forgot to mention there's not just pharma postdocs, there are academic postdocs as well, where you will also still get a lot of this experience. But those you will get paid a lot less, which is fine. And so there aren't really no limits to applying to a PhD only or a PharmD only as a PharmD, PhD, etc.
Gotcha. On the subject of the academic postdoc fellowships, do you think an academic fellow would have the same opportunities getting into an industry career as opposed to an industry postdoc fellow?
I think they'll have a lot harder time, honestly, as an academic fellow. It's because of the way the internal politics, I think, are. When you're an academic postdoc, you are fighting for your funding, right? You need to make grants. There's a different skill set that's involved. Whereas an industry postdoc, they've essentially budgeted for your project. So it's very project-oriented, project-based. I honestly think an academic postdoc is very rigorous. It's just, it also takes a very specific type of person to go into it.
Gotcha. So you really have to flex your transferable skills when coming from the academia side into the industry side.
Yes, very much so.
Perfect. Timo asks, will you go over QSP and MIDD with the group in a future session?
Very likely not.
What is QSP and MIDD, if you don't mind me asking?
Model-informed drug delivery. So this is actually right now a very, very hot topic in pharma. But it's also very, very, very new. And so it just as the title talks about, right? The model-informed drug delivery. It's saying, can you model the way that the drug is going to end up going through the body and use that for your application? And that's what a lot of people have been doing for a little bit more well-known mechanisms. And that's essentially MIDD. QSP, on the other hand, is like quantitative systems pharmacology. This one is like very technical. You're using like mathematical modeling, and you want to see how the drugs, right, will interact with any of your biological systems. So I personally don't see a benefit of doing a presentation on these two, only because they're not very beginner friendly, in my opinion. There are a lot more other topics within clinical pharmacology that should be gone over before even thinking about these two.
They sound very complicated. As a layperson to all of the drug delivery stuff, I would think, with MIDD, based on how you framed it, that would be more along the lines of something where you'd have a better idea of the actual mechanism, maybe like a sort of precision medicine instead of something general.
Yeah, so that's why these are going to be like your cream of the crop pharmacologists. And it's not very easy to make one of these models either. So for the purposes of, this Discord, I don't think it's a very good topic because it might just... I'm afraid it's going to end up veering people away from clinical pharmacology.
Oh, gotcha. Yeah, a very complicated topic. I could tell just from how you phrased it originally. So it's okay. We can move on. Howdy asks, how often do clinical pharmacologists and clinical scientists differ in their perspectives around things like dose selection? And when that happens, what does the resolution process typically look like?
I think actually not as often as you might think, because this is what people kind of talk about in industry, which is stay in your lane. When you are the clinical pharmacologist and you have an opinion on the dose, more often than not, you will be taken seriously because that is your bread and butter. That is what your job essentially, majority of your job, a lot of your job will entail. So if clinical scientists are arguing or have differing perspectives, you need to start asking them, why do you have a different perspective? What exactly are you trying to get at, right? Because maybe it's the higher ups that are telling them, or maybe it's because they just really, really, really believe in the efficacy. But that's only a part of drug discovery, drug delivery, right? You also have to think about the CMC aspects. For example, if clinical scientists are like, I want a liquid formulation, clinical pharmacologists and CNC, they have to tell you, is this possible or not? Because it might not be possible. And so you need to defer to your SMEs at that point. Your clinical pharmacologists are your SMEs for this. And so that's why the resolution process, and that's why is pretty smooth, and the differing in perspectives are not as common as I think most people would think.
All right, thank you so much. I believe we've got 2 more questions, if that's okay with you. FDA asks, is there a perceived hierarchy between PhDs and PharmDs working in clinical pharmacology, particularly regarding roles or career advancement?
So in the beginning, I think there is not a hierarchy, okay? It's more of a barrier to entry. They will look at a PhD sometimes more favorably than a PharmD, because the problem is some PharmDs don't know how to market themselves. They don't tell these PhDs or their interviewers what skills that they actually know. And the thing is, these PhDs don't know the PharmD curriculum. They don't know what you've done. They probably don't know what a rotation is, right? But once you get into the role, you are usually treated equally, because at that point, it's just your skills. Like, do you have the scientific and technical knowledge? Can you use these tools? And do you have the soft skills? It's not, I think that's the difference too with clinical scientists, is you're competing against an MD as a clinical scientist, and they will always have that leg up, right? They're an MD. Whereas for a PharmD PhD, the difference is, honestly, they overlap pretty well. So just get your foot in the door, and then work your way up from there.
Excellent. Well, Roy, I think that's the end of our questions. I just want to say thank you so much for taking the time to put this together and present to Biocentrica today. I know everyone in the audience was super excited to see this presentation, and I think it went super well. For everyone else, thank you for joining us. It was such an excellent session. I will keep everyone posted for future events as well. And Roy, again, thank you so much.
Yeah, anytime.